Hello, everybody. I haven't made a video in a little bit, so I thought, what the heck? I got kind of out of the routine of making a video for you guys in these first couple of served. I have, what is it, 10 slides that we've done so far. I didn't even talk about them, so what the heck? So I'm going back to it. I know we went through all of this, but I just kind of want to talk to you about it. So, in watching all of your flip grids too on the TED Talks that you picked, some really interesting stuff. And you guys did a really nice job with it. So, nice work, people. But biotechnology, when you look at biotechnology, you know how like I always start the beginning of a unit when I do the warm ups, like breaking down the word and looking at the learning targets and asking you about like what things look familiar to you. So when you look at biotechnology, what does bio mean? I hope that you're thinking, duh, Mrs. Nip, it's life. And then technology is something that we use to make life better. So you put that together, it's using living organisms to solve real world problems or to solve like a problem and make things better for people. Um, One of the big things, and I gotta move this out of the way again, with biotechnology and something that you have to understand is to do this, DNA is used. And a lot of times we're manipulating and changing DNA because then we want that DNA to be programmed to do something else or to be fixed. So it starts with cutting DNA. In nature, what we have found through studies is bacteria have these enzymes that cut DNA. They cut virus DNA to kill those viruses. So just like viruses, the coronavirus or whatever, influenza and Ebola and I don't know whatever other virus there is, it tries to take over our cells and then would make us sick. It does the same thing to any other living thing's cells, and even in bacteria. So it tries to invade it. And bacteria have these enzymes called restriction enzymes that we have found that cut DNA. So basically, it's going to chop that virus's DNA up so then that virus can't work. Why cut DNA? Scientists have learned to use the same enzymes to cut DNA at specific places. It's crazy to think how scientists can even figure that out and do this. It still kind of blows my mind about that. And it's like, phew, I don't, how can you like study that? How do you know? Um, but I mean, there are ways that they can figure it out. And if we were in class, we would have this. I have this really nice activity that we could do that would like show you that, and that you would use a restriction enzyme and then put it into place um, on paper, not the real thing. That would be even cooler. But anyway, so scientists have learned how to use those restriction enzymes from DNA to cut DNA in specific places, and they can now isolate genes for those studies and transfer DNA between organisms. So cut the DNA from one organism and then put it in the other. There's lots of examples when we talk about genetically modified GMOs. Those are all where a piece of DNA from one organism was put into another. And it's usually to make it better. How it works: restriction enzymes can read DNA and recognize certain sequences on where to cut DNA. This action destroys the virus DNA. So these restriction enzymes know to cut on a particular piece, segment of DNA. Hopefully you watched that video right there. How it works: it can also open up DNA in higher organisms, more complex organisms. If the same restriction enzyme is used to cut open DNA of two organisms, the ends of the DNA will become complementary. That means you can join the two organisms' DNA together. This is like an activity that we would have done with the human insulin gene in class. Dang it, coronavirus. Recombinant DNA. So it's kind of like what the name is saying, recombinant, recombine. It's where they take the DNA from those two organisms and then put it together and that makes recombinant DNA. So it's DNA from multiple organisms can be spliced together. The same restriction enzyme is used for each organism. It cuts the organism's DNA at the same sequence so then it can be joined back together. If I move this right here, so this is the double strand, this is DNA. It's cut at a particular site on this DNA right here and this other piece of DNA right here. And then you can see that they will join perfectly together. Yay, recombinant DNA. They call those ends sticky ends. When the DNA cut is an offset pattern like this, kind of looks like a step almost, it joins together. As long as the other DNA is cut in the exact same sequence, those sticky ends will join together. Here's a gene maybe to make insulin. So we know insulin. Hopefully, you think of people that are diabetic. People that are diabetic, their bodies typically don't make insulin. Depends on if it's type 1 or type 2. Hi, Hannah. Um, but their bodies typically can't make it or can't make it very well. So where do you get the insulin from? It's not like I can just donate insulin to some people. What we have done is we have taken our human insulin gene, programmed and put it into bacteria. And if that bacteria has the human insulin gene, it reprograms the bacteria to make insulin. And then guess what? Then we can process and take that insulin. And now people that are diabetic get that life saving, medicine that they need. Pretty cool, right? Being inserted into bacterial DNA, and it's a circular piece of bacterial DNA. We call it a plasmid. And now you have a bacteria with so this came from this is like the human insulin gene being put into the bacteria, and it will reproduce and pass on its gene to more bacteria. And then you have a bunch of bacteria making insulin, which we again can use and process for people that are diabetic. It's called gene cloning. Every time the bacteria reproduces, since it has that new gene in it, it copies that new gene, and now we have a whole line of bacteria cells that will keep continuing to reproduce and have that human insulin gene in it, and that will keep making it. And now you guys are going to read about this, or not read, you're going to watch a video about this. It says the Flavor Saver tomato was the first commercially produced transgenic organism. Transgenic also means it's genetically modified. It's the same thing. Again, in science, we have to have, like 10 different words that mean the same thing just to confuse you all even more. But when you see transgenic, I want you to think, okay, that's genetically modified, meaning it got DNA from one organism and that DNA got put into where am I here? Another organism, and has that recombinant DNA. So it's genetically modified. And again, genetically modified, usually we are doing that to make a product or make life better in some way. So watch the video about this, and then there are some video questions afterwards, and then that's it. Hope you guys are all doing well and hanging in there. I'm kind of hanging in there. Okay, peace out.